Hi there. I wanted to do three kind of short parts on chapter 7, and then you kind of pick and choose what you'd like to go through. So the first portion here, just go through some definitions. I usually don't do a lot of lecture on this just because it's kind of dull to listen to, but chapter 7 in general is about microbial nutrition. And so we kind of have to think about what microbes need, and then we kind of can do the fun part, which is thinking about how to kill the microbes by sort of pulling things out of their environment or giving them different toxins that cause them to either take them up and then the cells are disrupted or they're oxidized, or lots of different ways we can disrupt bacteria. But right now, we're kind of thinking of the nutritional side of it. The second part of this series, I'll go into the gas requirement. So if you're taking lab in particular, that might be helpful to listen to. But the first part, I'm just going to go through some basic definitions. Nutrition is here. The idea of nutrients, macro versus micronutrients. Okay, so those are typically like your proteins, your carbohydrates, fats, micronutrients, trace elements. So a lot of times things that we take in our diet, like in a multivitamin, those are typically going to be more this real small amounts that are there. Depending on what it is, you might need more, you might need a lot of it. Some microbes can synthesize like some of their own amino acids. So, it depends a little bit on the microbe, like which macronutrients, but usually there's a selection of micronutrients that most organisms need to grow, things like iron, zinc, things like that. So you do not need to know, so I'll put don't memorize the chemical composition. I simply put this here. I simply put this, I'm just writing this down. I simply put this here just so you can just kind of just get a general feel for what the cell is made up of, kind of break down this idea into, your macronutrients, right? So here's a lot of your macronutrients. Okay, and this is, just like our diet, so nothing really shocking or different here. This is going to tend to be your micronutrients, whether that's phosphorus, whether that's sodium, whether that's calcium, all those different things that we take in our diet. I don't ask you the percentages or anything like that. It's just to kind of give you a view and more of a visual of macro versus micro. Most of the microbes that we focus on in this course would be heterotrophs, meaning that it has to take in carbon normally from somewhere else. Often that's you, right, if it's a pathogen. Autotrophs, those are like your photosynthesizing organisms. We do talk a little bit about algae, so they would fall into that group there. But most of the time, we're worried about heterotrophs, right? Because they're the ones that are after you. So this would be pathogens, but even the good organisms, right? Even the beneficial microbes. We are heterotrophs, right? We take in our carbon from somewhere else. Saprobes, which break down dead or decaying matter. So all of those, and some of those can be quite beneficial to, the existence of the microbes. The nitrogen and oxygen, not going to say a whole lot there. We'll get into more into gas usage later, but just so you know that, those are critical. Nitrogen is a big part of energy generation, right? We talked about energy generation, but also the nucleotides as well. Oxygen tends to be important, although keep in mind some microbes can't tolerate what we call free oxygen, right? This is the free oxygen, the O2. But even if they are what you call obligate anaerobes and can't tolerate free O2, oxygen is still bound up in other molecules in the cell like carbohydrates, lipids, and nucleic acids. Okay, so hydrogen is just, a sort of a binder to carbon. Helps keep pH in the cell. And then now we're getting into some more of these kind of micro type nutrients here. So micro, maybe I should put nutrients because you're going to think that means microbiology. Micronutrients, much smaller amounts of these are needed. And these are still needed in pretty high amounts, phosphorus and sulfur, because they make up things like DNA and protein. So they're still pretty important. So growth factors, those are just things that are taken in the cell. Typically, it's going to be something like an amino acid or something with some other nitrogen-rich compound that has to be taken in for the cell to survive. Again, some microbes can make their own amino acids. I mentioned saprobes are decomposers. And they'll break down, even as some organisms will switch, they'll kind of be a pathogen on the living host, but as the host is, the tissues are dying or becoming necrotic, they'll actually start feeding on dead tissues, and then when the organism dies, they'll break down the tissue. So they play dual roles there. You're not necessarily stuck in one or the other. A lot of these use enzymes, and if you remember, we talked about enzymes, so a lot of times when we say they're releasing enzymes, hopefully you're thinking about the term exoenzymes, right? They're digestive enzymes that are secreted outside of the cell. The oh, there we go. Another way to look at that, we've talked about exoenzymes, but just that idea of secreting outside of the cell. And so here is, on the left here, digestion of bacteria and fungi. Here's the debris it needs outside. It's too large, right? So it's too big. So it secretes these enzymes. We said these are exoenzymes, right? Because they're leaving the cell and going out into the environment. Then when things are broken down, then they can be ingested or ingestion. Just like, it's kind of the idea of like when we chew our food, right? We can't necessarily break down a whole piece of most large particles of food, so we chew it to break it down. Well, microbes don't have teeth and all that complexity there, but they can secrete enzymes that do that for us. Look at the term, the idea of parasites, that they are unable to grow outside of the cell. Some pathogens are parasitic, but they can also be free living as well. So there's the difference between obligate parasites, and the obligate means you're like committed, you are obligated to live that way. So some organisms have to or can only live parasitically, where other organisms can switch, kind of like that idea between switching between being like a pathogenic parasite and a sapro. We talked a little bit about how nutrients often have to be absorbed. Sometimes they can just be pumped over the cell membrane in larger forms, but typically they have to be broken down or be small enough in the environment. And you're going to have, when you look at the cell membrane, you're going to have transport proteins, right, that bring things in over the cell. And sometimes those are active, sometimes those are passive, depending on whether they need energy or not to bring things over. Some things just, again, diffuse over across the cell, like some gases and things like that, but a lot of times it has to be moved over actively. Review the idea of osmosis, of water moving across the cell membrane. One of my favorite pictures probably in this chapter is just how different bacterial cells respond to watery and kind of non-watery environments. So isotonic, which we know is hardly ever really exists, usually the cells doesn't have enough water, has too much water, but isotonic, you have this sort of even flow into and out of the cell. Okay. But in a hypotonic environment, you have a lot of water flowing into the cell. I'm sorry, I think I said hypotonic, so hopefully I did. So a lot of water is going in. Now what they don't always show here is you do still have a teeny bit of water coming out, but the bulk of the water is moving into the cell. And one of the ways we capitalize on that when we are treating infections is that we try to weaken the cell wall. So I've talked a lot about cell wall weakening antibiotics like the penicillin classes, methicillin, ampicillin, vancomycin. When that wall is weakened, water is able to move easier into the cell and build up. And then eventually it can cause rupture in a lot of cases. So the cell wall will protect it if it's intact, but at a certain point, if you can weaken it, so you can enhance lysis by weakening the cell wall. Okay, and we also make a substance called lysozyme, which weakens the cell wall. So we also do that naturally. Now down here, these are of course your gram positives and your gram negatives, okay? But down here you have your wall-less bacteria. And so one example we talked about were the mycoplasmas. The mycoplasmas, the water can move into the cell quite easily. So they actually rupture really, really easily. The problem is sometimes cell wall weakening antibiotics are given and they don't really have the desired effect there. So they do have to work hard to pump water out of the cell. They have mechanisms to do that. So cell wall and degrading antibiotics aren't really helpful on these, but they are helpful on the ones with the wall. The other way we actually not necessarily kill bacterial cells, but limit their growth is by putting them in hypertonic solutions. And those are very salty or very sugary. That's one, think of it. You can also think high caffeine, anything that's a high concentration outside of the cell. I usually go for salts and sugars because that's something we can relate to feeling like after you have something really salty or thirsty, you can almost feel that water being pulled out of your cells. So it won't necessarily kill the bacteria, but it will dehydrate the bacteria, and it kind of prevents it slows down growth. When I say growth, not that the cell is getting bigger, but that it's able to grow in population. So when we look at things like potato chips, dried fruit, beef jerky, anything where we've put the food and we've added a lot of salts and sugars, yes, it tastes delicious, but one of the actual real honest to goodness benefits is that it prevents or slows the growth of microbes. If you were to rehydrate by flooding that with water, or could somehow remove the salt or sugar, there would probably be low enough levels of microbes they would start to grow again. And we do see that on occasion in foods we don't necessarily think of as normally having microbes growing. Sometimes they do. With the wall-less bacteria, they're just going to, of course, shrivel right away. And they're both shriveling. But if you look at the bacteria with the cell wall around it, it's got a little bit more protection there, but again, it's not going. Neither one is going to be actively. Doesn't necessarily kill these types. It just inhibits. Okay, and that's the idea with food preservatives a lot of times. We put a lot of stress on it. Sometimes it's a pH, right? With an acidic pH, like a vinegar addition. Okay, so that is it on those. I will do a part 2 where I'll move on into things like oxygen, but spend a little time just kind of thinking about this figure, thinking about water movement. I think the nutrition items, review a few of the terms like saprobe, parasite, but in general, I think you'll have a pretty good idea because most folks have a pretty good sense of nutrition and macronutrients and micronutrients. Okay, that's all I have, and I will pick up on part 2.